With that, I'd like to move on to introducing our final speaker of this group, who is Dr. Shetty from the National Prion Disease Pathology Surveillance Center in Case Western Reserve University in Cleveland. And she is going to be talking about her grant entitled Examining Copy Number Variants as a Possible Phenotype Modifier in Prion Disease. Dr. Shetty.
Thank you. Good morning, and thank you, Dr. Watts. He's a fellow Canadian. I'm a Canadian too, and it's nice to see more Canadians around. For some reason, we are all coalescing with prion disease, so it's good. And I also would like to thank my first two presenters, our presenters, because Brad gave me 2 extra minutes. So thank you, Brad. And the first presenter made my presentation even easier because he covered some of the things that I would have gone on and on and on, so now I don't have to talk a lot. So thank you very much. So I have nothing to disclose. I have no conflict of interest. No companies pay me to give this talk. That's what it means. This is just a reference slide because we geneticists tend to use a lot of jargons, so I wanted to make it easier for the non-genetics crowd here. So please go back to this slide if you have time. Most of the individuals here, you are aware of PRNP gene, that it causes genetic prion disease. So far, we know that there are about 70 mutations that are responsible for genetic prion disease, and I'm sure more are going to be discovered as we see more families, and we have a phenotype associated with it. And the way this is inherited is you just need one copy mutation. Genes come in pairs. You need one mutation, and if that's inherited in that form, it's called as dominant. It's called autosomal dominant because if you saw Dr. Knight's presentation, he mentioned PRNP gene is on chromosome 20, so that's an autosome. So 1 through 22 are autosomes, X and Y are sex chromosomes. So this is an autosomal dominant pattern, how it's inherited. So the first presenter already mentioned codon 129, so I don't have to talk a lot about it. In terms of, we know that the causative agent is PRNP gene, but what are some of the modifiers? Why do we see variability in the phenotype? Like a person who carries E200K, it runs in the family, but you see difference, age of onset, penetrance, or the variability. Why is it happening? Are there other genetic modifiers? So we know that in subset of white population, codon 129 is important, and in one of the Asian populations, codon 219 is important. Okay, so why did I pick copy number variants? One of the reasons is we know that in younger, like pediatric population, copy number variation. What that means is, if you look at our DNA, you'll either have deletion of certain regions or duplication of certain regions of the DNA. And I will tell you, we all carry this. It's normal. As a part of evolution, it's important. So it's like, millions of years ago, you had a tail and you didn't use it. You didn't need those genes, right? So they're going to become pseudogenes, and at some point you lose it. So it's a part of evolution. It's normal. What I'm looking for is that rare CNV that's not present in most of the people, but it will enrich in a population that is presenting with a disease phenotype. Okay, so CNV results in gains and losses. What does that mean? Loss means loss of the genetic material or genes, so that in turn would affect protein expression. So similarly, if you have gain, that means you have too much of that gene, you're going to have more expression, more protein, so again, that's not good. You want a balance. In the context of Alzheimer's disease and Parkinson's, CNVs have been well studied, but we know very little about it in the prion world. Like I mentioned, we know about CNVs in the pediatric population, especially in the autism spectrum disorders, schizophrenia, bipolar disease. But what we don't know, and I'm glad that Brad brought this up about the organoids, because it's very difficult to study brain. Like, if I want to study what's happening in the brain in terms of CNVs, no clinician is going to do biopsy for me for every, if I want to do research. So this is a great way of testing the brain genes. And something that based on the literature, we know that in the brain cells, so when it comes to prions, neurons are very important, right? It is that cell that deteriorates over time and then the disease sets in. What we know about these copy number variations is in the neurons, they are present in mosaic state. What that means is if I looked at 100 neurons, about 5 to 30 cells would have this variation. The rest would be normal. So it's a mix of normal and a variation. So we know about neurons in that context. And these neurons are fully differentiated cells because they have a high function order, right? They have to do a high functioning thing in the brain. So they're already differentiated, they're not dividing. So if there is a problem with these neurons really depend on the DNA repair mechanisms to repair it. But with age, our DNA repair mechanism deteriorates. So that is a problem for the neurons. So going back to the mosaic state of CNVs, like neurons are present in mosaic state, even the best of technology, and I'm sure Brad will agree with me that it's not easy to study mosaicism because there is no way for me to predict if the cells are going to be at 5%, 30%, or 1%. It's very difficult to study mosaicism. One thing we know about the brain is the largest part, like the cerebrum, the cortex, there are about 60 billion glial cells. So the glial cells are the supporting cells for the neurons. So you have the neurons and the glial cells, and they're present in billions. And if you look at the, is there a mouse? There is no mouse, okay, sorry. So if you look at the structure where it says C at the bottom, that is the cerebellum. So it's just 120 grams in size. It's a very small organ, like in our brain. But the way neurons are packed in there, it's almost 4 times compared to the cortex. So it's really present in a high population in the cerebellum versus the cortex. So because they're present in such a high proportion, our chances of finding that mosaicism or that cell line with the mosaic population is much, much higher. It's almost 4 times higher if I studied the cerebellum. That is my hypothesis. Let's see if it holds water, okay? And then if I want to study germline, that's not something that happens as we are aging, but something that happens when the conceptus was formed, we use blood to test that for germline mutations. So there are 2 questions. Why are these sporadic CJDs, like the VV1 subtype and sporadic fatal insomnia, why are they showing early age onset? And what that means is when something shows up earlier, that means it's behaving like a genetic disease. Because typically you're supposed to see this in the 60s or 80s, but you see onset in about 30, 40 years age old people. But some CJD, I have a patient who's 13 years old, 1-3, not 30. So why is it happening? And the second thing is same mutation behaves differently within the same population. Why is that happening? Are there other genetic modifiers? Okay? So I'm not going to go through this because this was something I put it out for the grant. So there are 3 specific aims. Same thing, try to understand CNVs in sporadic disease, genetic disease, and the third thing is understanding the function. So if there is gain or loss, how is the protein, what is the protein expression? So that we do using immunohistochemistry or Western blot, okay? And this is the technology I use for studying copy number variations. It's called as microarray technology. What you do is from the brain cells or the blood cells, you extract DNA, and it goes through this 4-day process of digestion, ligation, purification, fragmentation, labeling. I'm not getting into the details of the technology, but it's like a long process. And once that happens, the readout I get, this is what I get. So this is for every chromosome, I get to see this. So if you look in the center, where it says the blue line is at number 2, because we have 2 copies of all the genes or the DNA or the chromosomes, so that's normal. But anything that deviates from 2, whether it goes above 2, that means it's a gain, and below 2, it's a loss, okay? And at the bottom where you see all those dots and waves and all that, what that's telling me is, if you have a gain or a loss, can I tell if it is mosaic or not? So these two pieces of information that I get, it helps me understand if there is gain or loss and if it is present in mosaic state, okay? And this was my cohort of patients. So I looked at 19 patients so far, 35 regions of the brain, that was frontal cortex, occipital cortex, and cerebellum. And the age range is anywhere, it's between 13 to 75 years old. And I also studied 7 patients for germline. So I used blood, DNA. So this is the group of patients I studied. Majority of them, for this first set, I wanted to make sure I understand in the normal non-prion brain disease, these are not normal, but non-prion disease, are there CNVs and what type? So most of the cases, you can see that out of the 19 cases, I studied 7 just for the non-prion disease, so I get an understanding of what the baseline is. Okay, and then I have the sporadic CJD VV1, sporadic fatal insomnia. For now, I have just analyzed E200K cases, but I'm going to do the FFI and GSS as well. So the preliminary results so far, this is not what a scientist hopes for, like something is standing out. Right now, nothing is standing out, but it probably is coming, so we'll see what happens. So this is just the preliminary data. Out of 19 cases, 17 were absolutely no rare CNVs. So when I say no CNVs, I mean the ones that are not seen in the general population. I saw a lot of CNVs pretty consistently that we all carry. It's there. That's not the one I'm looking for. I'm looking for something that enriches in the abnormal phenotype or somebody who carries the mutation or has the disease. But 2 patients, interestingly, so when I was analyzing the data, I had no idea what subtype I was looking at. I didn't know it was sporadic, whether it was genetic, it was blind. So I was just going through, like you saw those outputs, I was just going through each and every chromosome, which is very calming. I kept doing that, and after I did the analysis, I said, oh, there are 2 cases. It's not showing small CNV, but it's showing gain of the entire chromosome X. I didn't know, I knew these were females because the data tells you if it's a male or a female. So 2 cases, and I don't know if it's coincidental or not, but they both turn out to be E200K MM cases. I'm not jumping to any conclusion, but I thought it was interesting. So first case, like I said, they were both females. Age of onset on the first case was 57. She had a disease course of 8 months. I just studied the cerebellum in this case, and this is how it looks. So on the left, my left, your left, yes, okay. It's always difficult to remember. On the left is the normal, like you see, the waves are at 2. That means there are 2 copies of chromosome 1. And on the right, can you all see the shift? It's gone above 2. So that's telling me there is a gain for X chromosome. But this is in a mosaic state because it's not gone all the way up to 3. It's hovering between 2 and 2.5. Same thing for the next patient, 66 years old. Disease course was very short, E200K, but in this case, I looked at the frontal cortex, occipital cortex, cerebellum, and blood. This is what the frontal cortex showed again. Can you guys tell, or am I just making it up? Okay, so there is mosaic for plus X. Occipital cortex has it. Cerebellum, see, like I said, my hypothesis about I should see it like, it should be very obvious in the cerebellum. I think it's there, but maybe it's not in the same proportion, even though the number of neurons in the cerebellum is much, much higher. In blood, again, it was difficult to see. It is there, but it's, I think for a trained eyes, because I've been doing this since 2005, I can tell, but I think it will be very difficult if you didn't know what you're looking for. So the first question is, gain of X, is this normal or is this abnormal? So one of the things that happen with females is, as we age, we either gain or lose X. On the other hand, males, as they age, they can lose their Y chromosome. It has no role, right, as we age. So we can lose that. Sex chromosomes are lost as we age, so it's a common phenomenon. So the question is this an incidental finding? And the other thing we know about the neurons is if there is loss of X chromosomes, you can lose neurons. So basically neurodegeneration. So is that happening? In that case, does that mean if you have a gain, is it protective or not? I don't know that because X chromosome also undergoes X inactivation, so it makes it very complicated to understand what it could be doing. So it's important to see this in many patients. So this is work in progress. I have 21 cases that I'm going to go back and analyze, at least 63 regions of the brain, and less negatives this time, like or non-prion disease. And if I find rare CNVs, then I'll be doing the functional studies. You guys, everyone in the room, I think you have seen these, the summary slides. I'm not going to go over it. I want to thank the Robert Vitanza Memorial Grant contributed by Michael Vitanza. Thank you very much. Without support from this grant, this work wasn't have been possible. So thank you very much, and CJD Foundation. And I definitely want to thank Dr. Appleby, because when I ran this idea by him, he's like, sure, Shashi, go for it. Like, so, that kind of support is very important when you're trying to do something. And of course, my team there, who have been asking me to keep a smile on my face, it wouldn't have been possible without you. So thank you very much.